All right, here we go. Let's jump right into this. We are talking about muscular systems still, and this is going to be part 2 for our muscle system lectures. We're going to insert a little bit of nervous system anatomy in here as well. So we're staying on the same overarching theme of macro and micro structures. We're probably not going to go anywhere past that because I don't want to confuse everyone too much. But once we start getting down to the smallest levels of muscle cells, we want to start talking about the relationship between the nervous system and the skeletal muscle system, excuse me. So let's talk a little bit about the anatomy of nerves. So we have motor neurons, all right, and this is typically what we're referring to as a nervous system cell with reference to movement, okay, because a lot of our nervous system cells are connected to a lot of our muscle cells as well. And this is how we get some signals from the nervous system that come, from the ascending pathways from the brain down the spine into those motor neurons. And then that signal goes to the muscles, and that's when a muscle knows it's supposed to contract because it's being signaled. So we have motor neurons that contact muscles through the surface of the muscle cell. And on the surface of the muscle cell, we have a specific area called the motor end plate, where that signal is going to terminate electrically, and then it's going to become a chemical signal, and then it's going to make a signal on the muscle cell. And this is what we call the neuromuscular junction, right? So just like a freeway or multiple freeways come together, this is essentially an area where multiple systems are coming together. We have the neuromuscular system, or the nervous system, coming into contact through the junction of the neuromuscular junction with the muscle. So when we're talking about the convergence of these two systems, what we have is a muscle cell that typically only has one neuromuscular junction. But the opposite exists when we're talking about it from the perspective of a motor neuron. A motor neuron is going to innervate many muscle fibers, right? So again, let's try not to confuse the two. A muscle cell is only going to have one connection with the nervous system, which is through that neuromuscular junction, that motor neuron that innervates it there. However, the motor neuron that innervates that muscle cell is also going to innervate many other muscle cells. So we would term this a motor unit if we wanted to give that a name. And a motor unit is going to be the one motor neuron that we're talking about specifically, and then all of the muscle fibers that it's going to send its electrical signals to, all of the muscle fibers it innervates. And so all of the muscle fibers that are innervated by that motor neuron are going to contract when that neuron is sending the electrical signal. It's extremely important. We're going to talk about it a little more in the future when we are specifically going over the neural system, but we refer to that as the all or none principle. So here is a basic picture of what we were just discussing. You have the muscle belly in red, you have the nerve cell in yellow with all of its little offshoots called dendrites. You have the nucleus, which we see in just about every cell in the center there, right? It's the little brain of the cell, the place that houses the DNA. We see this big, long protrusion that connects to the muscle cell. We call that the axon, specifically the yellow part, or the cell part is the axon. And then there's these little fat cylinders that wrap around called myelin sheaths. And in between these cylinders of fat, we have little yellow spots, which are still pieces of the cell, but that's essentially where that electrical signal is going to be jumping to make its way down to the muscle. And then we can see the neuromuscular junction, and we can see, again, the muscle that we talked about. So let's jump back to the skeletal muscle system again, and let's get a little bit deeper inside of the cell. So inside of the cell, we have all of the stuff once we've gotten below the sarcolemma, right? And all of that stuff we're going to refer to as sarcoplasm. In a typical cell, we call it cytoplasm, but we're going to use the prefix sarco when we refer to the muscle cell. This is also going to contain the contractile proteins that we're going to illustrate later on. It's going to contain other types of proteins. It's going to have some glycogen and fat stores, which are going to be for providing energy. It's going to have some enzymes, which are going to facilitate metabolism within the cell. And then it's going to have some specialized organelles like mitochondria, which we all love to joke is the powerhouse of the cell, but it literally is the powerhouse of the cell. And then the sarcoplasmic reticulum, which provides calcium for the entire muscle cell, which we've learned up to now is one of our main facilitators in contraction. Within that stuff, right, we have, again, those contractile proteins, but those contractile proteins are arranged in what we call myofilaments. And this is the majority of the cell. Okay, the majority of it is going to be of these contractile components, which is why it looks the way that it does under a microscope. And specifically what we're referring to are the actin and myosin. So here's a little bit of a closer picture. Again, we have the muscle sarcolemma surrounding the outside of our muscle cell. We have the sarcoplasmic reticulum. We have these T-tubules, which are connected to the sarcoplasmic reticulum. The T-tubules also make somewhat of an opening connection on the outside of the sarcolemma to receive a signal from the motor end plane when it travels down the muscle cell. We could see our myofibrils that we indicated were organized cylinders, longitudinal cylinders of actin and myosin. And then we could see our mitochondria, which are going to be providing the bulk of the energy, at least through oxygenated methods for our cell. What we see at the top right here is not indicated, but that's actually one of the nuclei of the cell. Okay, so let's get a little bit deeper. Myosin is we're going to take a look at what this looks like as well after this slide, so just bear with me and try to create a theoretical picture in your head until we get there. Myosin is going to have globular heads, it's going to have a hinge point, it's going to have a fibrous tail, it's going to be the larger filament of our myofilaments. The head protrudes away from that fibrous tail, and then a pair of myosin filaments come together to form what's called a cross bridge, and that cross bridge is connected to the other filament, which is the actin. So actin is then going to exist, like I said, it's going to be a smaller, a thinner, not as thick as the myosin filament. It's two strands, and they're arranged in the form of a double helix. So if you can imagine what DNA looks like, it's very similar to that, except it's packed a little bit tighter without the ladder rungs in between. Then we have the sarcomere, which is the unit that the myosin and actin filaments are organized into. And as I've mentioned a couple times, it's this longitudinal cylindrical arrangement of actin and myosin throughout the length of our muscle fiber. And there's going to be a way for these sarcomeres, because they're the individual units of muscle, to organize and connect themselves so that they can form the length of our myofibrils throughout the length of a muscle. And that is through, first and foremost, the adjacent myofilaments. Okay, at the center of our sarcomere, they're going to link at what's called the M bridge, and that's at the center of an area in the sarcomere called the H zone. Then we have the actin filaments aligning on both ends of our sarcomere at what we term the Z line, and that will illustrate itself once you see the picture. And then if we look at it from a cross-sectional picture, what we're going to see is that 6 actin are going to surround each myosin filament. And then if we look at it from the perspective of actin, we're going to see that 3 myosin surround each individual actin. So like I mentioned first, we have the H zone and the M bridge at the bottom of this picture, right? That's going to be where our myosin filaments, pieces of the filament, are going to link. And then if you look a little bit higher up in the center of the picture, we see the end of that cylinder with all the bands on it. That would be termed the Z line. That's going to be where our actin filaments are going to link to each other. And if we look at the hexagonal arrangement that we saw at the end of the last slide, it's going to be at the top of this slide in terms of the picture. At the bottom, I also want you to note some of those different structures we talked about with the actin and the myosin, right? We had that double helix-like structure within the actin where two strands rotate around each other. And then the myosin strand, being a little bit thicker, also has the tail and the globular head that in the picture at the bottom there, we do see an actual interaction with the myosin and the actin forming that cross bridge. Okay, so let's talk a little bit more about the appearance under a microscope, because that's going to be kind of important for identifying the differences between skeletal muscle and other types of muscle. So our myosin and actin, if you could see from that last theoretical illustration that we had, created bands, right? And typically they alternate light and dark based on the thickness of the band, and that's what gives it the striations that we can see under a microscope. We saw this all the way back in the very first muscle lecture. And then we could start notating, because of these different colorations in the bands, specific areas, right? So if we're looking at the A band, we're talking about the alignment of the myosin filaments. That's the center. If we're talking about the I band, we're looking at adjacent sarcomeres, and we're seeing only actin filaments, so kind of that Z disc alignment area. The Z line, as I just mentioned, is the middle of the I band, which is why that makes sense to go to it next. And it's a thin, dark line that travels through that I band, and it's longitudinally traveling through the I band. Then you have the H zone, which is the middle of the sarcomere. Remember, our M zone is right in the middle of that H zone. And it's only myosin present. And this is going to decrease when we actually see a muscle shorten. So that area of the sarcomere will actually come close to disappearing, I guess you could say, when we see a muscle actively shortening, because the actin is moving over the top of the myosin. So we're not having such a thick H zone anymore until it returns back to a normal length. And then subsequently we see the I band, which is where those Z lines are. That's going to decrease during contraction as the Z lines are going to be pulled towards the center of the sarcomere. So those are our two areas where we're going to see some shortening occur. Then we have the sarcoplasmic reticulum, right? What exactly is that for? So these are going to be a system of tubules that are surrounding each individual myofibril, and they terminate right at the Z lines. And believe it or not, that's where the T tubules come into play, right at the Z lines, and those connect to our sarcoplasmic reticulum. So it's almost as if the entire myofibril system is covered by these tubes. Think of it that way. This is essentially where our stores of calcium are going to be, and they're going to be stored inside of vesicles within the sarcoplasmic reticulum. And as I mentioned earlier and in the previous lecture, we know that calcium, the release of calcium and the binding of calcium, is what's going to control the ability for a muscle to shorten. So then the T tubules, that's short term for transverse tubules, are going to run perpendicular to our sarcoplasmic reticulum, right in that I zone over the Z lines. And they're going to be connected to the sarcolemma, like I had mentioned before. And when the nervous system sends a signal through its nerve cell down the axon, it's going to terminate, and then it's going to send a chemical signal across the neuromuscular junction to the motor end plate. And then that's going to send a charge down the sarcolemma of the muscle cell. And because of these T tubules, it allows that signal to simultaneously be sent down towards all of those muscle fibers, meaning that we can release calcium on all of those myofibrils, and we can get a contraction of all sarcomeres simultaneously, which kind of goes back to what we were talking about with the all or none principle. Looks like I'm a little delayed here. There we go. Oh no. Looks like that's it. All right. Well, thank you for listening to Skeletal Muscle Series number 2, and you have a wonderful day. See you next time.